I'm Michael Birk. I'm one of the new grantees for this program. And I'm going to give you guys a vision for quantum optics. What's quantum optics? The first part of it is, optics. We have light. This part, I think most of you are familiar with. And the second part is quantum. I'm going to explain what that is and what we've been overlooking. So what my vision is, this will clarify, as we move forward, is that there's some things with quantum optics that we've put aside for a long time, ever since we started doing quantum optics, depending on when you started counting, but let's call it 1970s, that we've been ignoring. We've been assuming, is this a laser pointer? Who knows? Well, we've been assuming that light is weak, right? Everything that's quantum is like, 1 photon, 2 photons, 3 photons. We've been using quantum systems that are few mode or single mode. What do I mean? When we say we have, 2 photons, we have one in one frequency or direction, one going in the other direction. So these are 2 channels. We haven't been doing things with like 1,000 channels. And the final thing is, personal pet peeve of mine, is that all of our measurement devices are bad. I'm going to get into this near the end. So what's quantum light? We have 2 types of quantum. The first kind is what we'd call?
Just the lasers right there.
The green thing. Okay, nice. So one kind of quantum that we have is superposition. This is the Schrödinger's cat kind of situation. The cat is dead and alive. What this means, the reason this is interesting, the difference between this and the cat is dead or alive, is that you can have something that interacts differently with dead cats and living cats, and if it interacts and you measure that thing afterwards, it will look different than if the cat was either alive or dead. The second kind of quantum that we have is entanglement. This will be a whole lecture if I try to define this properly, but what it means in principle is that you have systems, 2 systems at least, that have mutual information about the other. And if you want to know everything about one system, you might discover more, not by measuring it, but maybe measuring its counterpart or performing a joint measurement. So the joint information of the 2 systems is a lot more than would normally be reasonable. So we know how to generate quantum light. We do this kind of thing, right? We're taking 1 photon. This is the 2 omega. We're taking 1 particle of light. We're putting it into some physical system. This delivers a kick to the physical system, and this kick may cause the photon to split into 2. Right now we have 2 photons. Each of them has half the energy. We have energy conservation, or maybe one has 1/3, one has 2/3, right? But it has the sum, and we have momentum conservation. So these are entangled now. If you measure one of them, for instance, then you know the energy of the other based on conservation. But this is kind of a false picture, because you don't actually put 1 particle in and get 2 particles out. This is a very inefficient process. So you put 1 million particles in, and one of them will split. Right? This is a problem, right? Because what we end up doing is we have our quantum particles, and we do interesting things in the sense that entanglement is interesting, but we don't kick any further physical systems. We never drive physics with quantum light. We just do normal experiments. So we take these are our 2 photon source, or 1 photon. You put it in a beam splitter, which is like a semi-transparent mirror. You do some measurements. But what if we could use this quantum light to do interesting physics? We could kick some system and then see what happens. This is something that has not been looked at until maybe the past 4 years. And the reason that this hasn't been done is that quantum light is weak. You get this process that's efficiency is 1 in a million. What are you going to kick with, right? It's like I can't burn the table with this laser pointer, right? This is not strong enough. I can't cause any interesting physics. But this is not true, actually. So this is research that's been published this year that I took part in. It turns out we can generate very, very strong quantum light, quantum in the sense of superposition, not in the sense of entanglement, where we can take light. So this is, classical light. It's oscillating like a wave. and it has a very specific phase and a very specific intensity at any moment. We can actually generate light that's very intense and has some superposition. So this is a distribution of intensities. It goes from plus some maximum value to minus this value. And we can drive physics with this. So for instance, this is an electron on a band structure, basically the potential of a crystal. And, well, this is an electron, this is a hole. Usually when you shoot light at a crystal, you're moving around electrons, and they move around on the band structure. Now, if our light is quantum and it's strong enough to give this kick, to cause this interaction, well, we suddenly have a lot of questions, right? If our light is, a superposition of strong and weak and even like minus, right? So it's going in both directions at the same time. Is the electron going in both directions in the same time? Is it interfering with itself? We have this richness of physical phenomena that we don't even necessarily have the mathematical tools to look at yet. And so this is published, right? This is a barrier that we're breaking through right now. So the second part of my vision here of what I think needs to change with quantum optics is the entanglement direction, right? So we have two photons. We have interesting correlations. Now let's say you want to build a quantum computer. So how do you build a quantum computer? You need a lot of bits. And the magic with quantum computer is that you have correlations between all these bits. But our process is, let's say if you're making it out of light, if your quantum particle is light, you're making pairs, right? And you're making another pair. So these are correlated. These are correlated. You want all of them to be correlated, so maybe you put the middle two in a semi-transparent mirror. They're mixing. Now you don't know which is which, and now all four are correlated. Now let's say you want 1,000 correlated bits. So you need, 1,000 semi-transparent mirrors. This is basically what's going on here. This is a heroic effort. There was this 2020, there was a Chinese paper about how they did the quantum advantage. They beat Google's results. They have, each one of these lines is something that creates two photons. And then each one of these boxes is a semi-transparent mirror that does this mixing process. So this is not scalable, right? Let's say you want to do a Moore's law kind of thing where you multiply the amount of bits you have by some power every year or so. Even just in terms of manpower, this doesn't work out, but experimentally it doesn't make any sense either. And why is this? It's because we're trying to build a huge system out of single blocks. And the reason for this is that we've been shying away from complex systems, right? So the state of quantum physics right now is as if we had never invented quantum, let's say, statistical mechanics, and everyone was just doing completely classical mechanics, trying to follow where every particle is going. But the thing is, we have multi-mode systems. We have systems that naturally don't create just two photons, one in this direction, one in this direction. We have systems that naturally have thousands or tens of thousands of modes, right? We can go on the Thorlabs website, buy a multi-mode fiber. This multi-mode fiber has several transverse modes. That are being played off from the stage. It has different transverse modes that can travel in it. And all of these can interact. If you pump the light strong enough in the fiber, you get exchange interactions between these different modes. So this is naturally a system that has 10,000, 100,000 modes. It costs $100 per meter. And the problem is it's too complex, so we don't know what to do with it. Right? This is always the reason. The reason we start simple is because we know how to solve simple. But fairly recently, the classical world has also gone into this, like extremely multimode systems, and they have some interesting results. So it turns out that these cheap, extremely multimode systems naturally organize. Right? If you're looking at how much energy is in each of these modes, well, it turns out that while you're traveling through the fiber, you have random interactions, and what ends up happening is that everything kind of organizes. All the light wants to be in some distribution of modes. Kind of like how you take a lot of particles in boxes, put them together, statistical mechanics, and the lighter particles are always going to end up faster, and the heavier ones are going to be slower when the temperature equilibrates. So I've also done some work on this just as a not huge test. So just to explain what's going on here, this is, instead of 1,000 different modes, we have 20 modes. We start with some energy in two of the modes, and we find that they kind of organize in a decreasing manner, just naturally. Now, what happens if we look at the information correlations between modes? What we find out is originally there's no correlations, everything is black, so there's no mutual information. And as you travel down the waveguide in this case, there appears some kind of structure, and it appears naturally. So there's a case that this will also work for quantum systems. Finally, I'm going to steal 1 minute of the question time. Everyone who's ever done quantum optics will, within maybe the first 2 months of working on quantum optics, immediately become jealous of the people who are doing classical optics. Why is this? Well, you have your quantum source, you want to quantify all these superpositions and correlations, etc. You have different measurements. You can measure the probability of measuring some amount of photons in a given time. You can measure second-order correlations, like the variance of the number of photons, etc. And you can do these more complicated things that tell you everything about the quantum state. If you search any of them, in Google, you'll find research papers. Why is this terrible? Because if you're trying to do a measurement in classical optics and you look for a camera, you're going to find a website that sells you a camera. If you want a spectrometer, you find a website that sells you a spectrometer. This, in quantum optics, we just don't have devices. You don't buy a device, you buy a PhD student, and he builds a device for something like 1.5 years. So this is the biggest building block, right? Quantum optics is in a state where, we only have the camera obscura, where our devices are not good enough and we can't just buy one. And this is the biggest problem moving forward for quantum optics. I think this is like the instrumentation is the biggest missing block to move forward, both for high energies and for multi-mode systems. That's it. Thank you.
Thank you very much, Michael, and I wish you success in building more than 20 component devices. How high do you want to go? 1,000?
Hopefully just one, right? Just one multi-mode fiber. 1,000 modes, 10,000 modes, and that's it.
10,000 modes, okay,
Yeah.
that's quite ambitious. Okay, questions from the audience, please?
Yeah.
No, I have no background in,
just so they can get into the recording.
first, I have no background in quantum physics, and even in normal physics, I have only the background of a first-year biology student.
Okay.
So, what I'm wondering is if you see, no, I forgot the word. Like where you see the distribution of how much, what the word was when they're matching.
Entanglement, superposition, energy,
yeah, the
momentum.
entanglement, the heat map, I think it is.
Yeah.
Then you see it varies a lot. It varies a lot, and it's not uniform, even when you reach the so-called end state.
Yes.
Is that a problem in using it?
So I think ideally, right, it would be uniform. Ideally, what I would love to get is that I have, this bunch of modes, some n modes, and they're all entangled to each other in an exactly predictable way. What I expect to happen is that I get some distribution. It's not necessarily uniform, right? It might be some kind of Boltzmann distribution or something like this. So some modes will be more correlated than others, etc. As long as it's a lot of modes and it's robust, as in it keeps happening the same way, all I care about is 1 meter of fiber, then I'm fine.
And you don't need to know the exact distribution each time to use it?
I do need to know the exact distribution. My hope is, I don't expect that nature will give me exactly the distribution that I want. My hope is that it will give me the same distribution each time. And I will have to figure out how to use it.
Okay.
Hi, very nice talk, first of all, thank you.
Thanks.
So you talked about using a multi-mode fiber for correlating the modes inside the fiber. So as I understand, you're limited by the number of modes in the fiber, right? Yeah. So if you want to scale it up more to a higher number of modes, so you need to use optical fiber with another dimensions or another refractive index, or even so, if you can get more modes, as I understand, only part of the modes inside the fiber are correlated with each other, right?
So
how can you scale it up to more modes?
Okay, so in principle, there's no reason to think that only some modes will be correlated and the rest won't, right? Because there's always an overlap. And yes, it's true that to get more modes, I need to scale the fiber up. But I didn't talk about this, but you have, in a multi-mode fiber, you have, your core diameter, which gives you more transverse modes. But you also have the frequency or time axis, right? So you can always use broadband pulses, and this will give you another dimension to get multi-mode. You can, let's say, multiplex across frequency and across spatial dimension. I'm not saying that, necessarily we will end up with one multi-mode fiber and this will be the solution to everything. But I do think that a multi-mode fiber can give you more modes than we can handle in the next 10 years, right? 100,000 modes is not that big of a number for a multi-mode fiber.
Okay, thank you.
Okay. So you have plans for the next 10 years. That's really good. Yeah.
So what are specific applications you can think of for this multi-mode fiber?
Okay, so for optical quantum computing, every branch of quantum computing, every kind of physical design of the computer, if it's superconducting or cold atoms or what have you, each one has its problems. For some, the operations, the gates are the problem. For some, quantum optics, the preparation is the problem. So for quantum optical computing, the big problem is this giant grid that I showed you before. Basically, if you want to prepare a large state, you have a lot of generators of these photon pairs. And each photon pair, it generates with some probability p, right? Like you turn on the device, and it tries to make 1,000 qubits. Each one of these qubits, it succeeds with probability p. If you have 1,000 of these, then your probability of success for just starting is p to the power of 1,000. So if p is not 1, you're out of luck. What I think is the path forward in this kind of case is exactly a multimode fiber. You have a multimode fiber, it creates naturally 10,000, 100,000, whatever, correlated entangled modes, and I think this is a very, very good quantum state generator, a massive quantum state generator, as long as we can find a way to make it robust, to make it happen the same way each time.
So hi everyone. Today I'm going to share with you a little bit about my research about enhanced nanobubble mediated BBB opening using nanobubbles. So, okay. First of all, the motivation for my research, even though all the things I'm showing today were done on healthy mice, is glioblastoma. So glioblastoma is one of the most aggressive brain cancers. It has life expectancy short and high recurrence rate, and the current treatment is a resection tumor when it's possible. It's not always the case, following radiotherapy and chemotherapy. One of the reasons it is so hard to treat glioblastoma and neurodegenerative disorders in general is the presence of the BBB. So the BBB, or the blood-brain barrier, you might remember from my previous talks, is a sort of a filter that prevents the passage of materials from the brain to the blood and vice versa. And in normal condition, it really helps the brain to function. But in the context of drug delivery, it prevents the treatment, and it becomes an obstacle. So luckily, we have a technology of focused ultrasound with which we can open the BBB for a certain amount of time and allow the drug delivery happen more easily. So all this magic of BBB opening is happening thanks to microbubbles. Microbubbles started the way in the ultrasound world as contrast agent, meaning they enhance ultrasound imaging. They compose from a lipid shell with gas core, and they about a fifth of the size of a red blood cell. And the interesting part is that when we apply focused ultrasound on these bubbles, they oscillate, meaning they periodically expand and contract, and they apply forces to their surrounding. And in case we apply the focused ultrasound in the brain, in the treatment area, the bubbles apply forces to the endothelial cells that compose the BBB, and make the BBB in that location permeable for several amount of time. However, there are still limitations when we're working with microbubbles. First of all, they are not so stable, meaning we have only about 30 seconds from the moment we inject the bubbles into the bloodstream to the moment we need to apply the focused ultrasound. And most importantly, as I'm showing here, the BBB opening is influenced by the diameter of the blood vessel. We show here in this graph that when the vessel diameter is smaller, the less BBB opening happens. And we want to enhance the effect of the BBB opening to make it homogeneous and maximize the effect, especially in the small vessel that compose the brain. And for that reason, we started to work with nanobubbles. Nanobubbles are also contrast agents, and they are much smaller than microbubbles. They also oscillate upon focus ultrasound application, and they have many advantages. First of all, they are more stable, they penetrate better into tissues and tumors, and we wanted to see if it also improves the BBB opening. So in this research, we wanted to see whether nanobubbles are better for BBB opening than microbubbles. So we performed the BBB opening in healthy mice using microbubbles or nanobubbles, and we compared the results using microscopy and histology. And the short answer is yes, nanobubbles were better, but let's dive into the details. So first of all, in the course of the experiment itself, we positioned the mice above the ultrasound transducer on this agarose pad, and we fixed his head with the hand holder. And he was lying above the transducer of the ultrasound that was in the water bath. And in the course of the experiment itself, we started with bubbles injection and focused ultrasound application, meaning we opened the BBB. And then we injected Evans blue dye. It's a dye that fluorescent in red. And after 18 minutes, we injected another kind of dye called fitsidextran. It's fluorescent in green. And then we continue with the tissue analysis. So first of all, we can see here, when we look at the whole slice of the brain, the red fluorescent, the Evans blue, it's a molecule that's supposed to extravasate into the brain only if the BBB is opened. So we can see here in the microbubble and the nanobubble group a red spot in the right hemisphere where we focus the ultrasound to, while we don't see in the control group such a spot. But if you remember, I talked about another dye, the green one. So the reason we use it is because we wanted also to be able to see the blood vessels themselves. At the bottom line, we wanted to quantify the extravasation at a resolution of a single blood vessel. So we wanted to be able to locate the vessels and then to calculate the extravasation around them. So first, you can see here in the middle column the red dye of the Evans blue and the extravasation from the blood vessels outside. And you can see in the green column, for each of the treatment groups, the location of the blood vessels. Okay, so regarding the calculation, I'm not gonna talk about all the process due to time constraint, but really shortly. As I said, we created sort of a mask of the locations according the green channel. We also implemented a machine learning algorithm that enabled us to do this process much more rapidly and analyze much more data. And then after we had these locations, we could use this information on the red channel and then go over around each vessel and have a measurement of the extravasation. We measure the median red intensity in the area around each vessel. And when we compare the result, we compare these median red intensities across different size categories for all the treatment groups. We can see in all categories that there is significant difference between the control group to the treatment groups. But the more interesting part is the comparison between the treatment groups themselves. So in the larger groups, there is no statistical difference between the microbubbles and the nanobubbles groups, while in the smaller one, there is a significant difference, and nanobubbles extravasate. In the nanobubble group, there is more extravasation. And when we compared, when we measure the improvement, it reaches 13% in the smaller diameter, while it diminishes while we look at larger ones. So in conclusion, we showed that using nanobubbles improve the BBB opening in small vessels without compromising on the BBB opening in larger ones. And our machine learning algorithm enable us to analyze much more data and have more robust results. And hopefully, we wanted to use this platform to better open the BBB, specifically in glioblastoma in other brain conditions. So thank you for your kind attention, and thank you for my lab members that helped me. And thank you.
Okay. Thank you, Roni. Questions? Please.
Great talk, thank you. My question is, if you open, maybe I'm unaware, but if you open the BBB, isn't there a chance of like harmful molecules to also penetrate the brain? Not only drugs and treatment?
Yeah, thank you for that question. The BBB is open for like a really certain amount of time, like a couple of hours, and molecules can pass, but still like not very large ones. And for now, we also like seeing studies of safety and everything, and there is no damage. Even for clinical trials, when they do these approaches and several times, they don't see any damage for the long term.
Hey, great talk, thank you. So I have 2 questions. So I noticed that the nanobubbles size is actually smaller than average cells. Can it maybe be used also to enhance the delivery of molecules inside cells, or only kind of used to separate different cells apart, like in the BBB? And the second question is also, I think that many times, for instance, in the context of tumors, even like not inside the brain, there is a problem of reaching within the tumor. So if we deliver these bubbles, maybe we can use them to enhance the delivery of chemotherapy agents or something else inside the tumor, like to separate the cells?
Yeah, so thank you very much for this question. So the first one was about drug release, right?
So the first one is whether we can use the bubbles to infiltrate cells and
intracellularly. I think so. I'm not sure right now. But they also can be used for drug release. You can encapsulate, not necessarily in nanobubbles, but in other kind of bubbles. You can encapsulate molecules that you want to deliver, and then you can release them near the area you want to deliver it to. But it sounds like a great direction. And the second question was about enhance the delivery of chemotherapy. Yeah. So first of all, another advantage of the nanobubbles that their size. Around tumor, there is an effect that's called the EPR effect, that the area of the tumor is more leaky, and we can see in the lab that when we inject nanobubbles into the bloodstream, they accumulate near tumors, for example, breast cancer that we do in mice. So this like an innate property of the tumor itself. And also you can create bubbles that have linkage proteins that link to a specific tumor and stuff like that to enhance the leakage and the targeting between them.
Okay, Maybe we'll take one more question, then the rest will take for discussion because we have to keep on time. One more question. Okay.
You might have addressed it, and I didn't notice, but what happens to the bubble after after you stop using them, or if they decay naturally, what happens to the materials afterward? Because at the bottom line, you are inserting on the material into the brain, and I assume unless your body can use it, it needs to get out somehow.
Thank you very much. It's a great question. So, as I said, because the microbubble started as a contrast agent, it's a material that is biodegradable, meaning they use it for ultrasound imaging like before the application, like the therapeutic applications. So we know it's safe, and they use it even for patients, like even before the therapeutic applications. And the microbubbles, I didn't mention, they already FDA approved, so they're safe and they are degradable after that.
Okay, toda Roni.
Thank you.
Okay, hi everyone. My name is Alon. I'm doing my PhD at the Hebrew University in the biophysics lab of Nathalie Balaban, and with a joint supervision of Professor Oded Agam, who is a theoretical physicist. So, okay, great, thank you. Okay. So, as a physicist who's trying to study these biological systems, then we quickly reach a very big obstacle. And that is the fact that a cell is an incredibly complex machine. It has thousands of interacting components, which makes reducing it into these minimal models that can be formulated mathematically usually almost impossible. And I think the main goal or the main challenge in my field in general is to find these universal patterns in these simple structures that we can actually understand in a more mathematical way. So in my group in particular, what we are interested in is bacteria under stress. And there are many different types of stress. You can think of environmental conditions that are not favorable to the cell. And there are many ways in which the cell responds to these environmental conditions, but we observe mainly two patterns of response. In one, you can think of a case in which all of the cells are kind of being driven into this one point that is very well defined and they are all pretty much doing the same thing. And we call this a regulated response, which is a well-defined and some kind of adaptive response to the environmental conditions. But there is another way in which the cell responds, which is, you can imagine it as a more chaotic, random-like response, in which every cell kind of does something differently. And this is kind of depicted in this image as these cells being spread apart in this landscape of possible responses. And we call this the disrupted state. It is something we are focusing a lot in our lab. And you can think of it as an unadaptive or sort of random response to stress. And why is this even interesting? Why do we even care about these disrupted cells? And the thing is, you can imagine an experiment in which we grow bacteria, expose them to stress, and then after a certain time wash away this stress and allow the cells to regrow. Now, what is very special and very unique about these disrupted cells is that the distribution of lag times, or the distribution of times it takes for cells to return to growth after the stress removal, follows this very wide distribution, in which you can see that even though most of the cells start to grow quickly, there is a very long tail in which bacteria apparently show up and start growing again. Now, what would happen if we treat this culture with antibiotics? What we get is that it is very common for antibiotics to only kill bacteria that are actively growing. And what we get here is during this finite time of the effectiveness of the antibiotics, it manages to kill the vast majority of cells, but then we have this small group over here that suddenly appears after more than a day, and these cells are able to evade the antibiotics and eventually cause the infection to regrow. These are called persister cells, and you can easily imagine why they are a very major problem to deal with clinically. So the goals of my research are, first of all, to provide a more microscopic picture of what is going on inside the disrupted cell. This is something that is very not well understood in the community. And I'm trying to look for these universal properties that take together all the different types of responses. As I said earlier, this is something very general. It's not something that happens only as an application to a certain type of chemical. So, but the main message is, I think that biology is incredibly complex, but this disrupted state somehow kind of peels away the layers of complexity and reveals these universal properties of cells, which are actually interesting to study on a more fundamental level. And okay, so to understand how we formulate cells mathematically, first we have to think of what defines a cell. And for example, you can think of what distinguishes this neuron, for example, from this muscle cell, even though they share exactly the same DNA. And the answer is gene expression. Gene expression is just the amount of RNA molecules that are expressed from every single gene in our DNA. And we can write it as this vector of integers, where every component of this vector represents a gene, and the numbers are just counts of molecules. So these vectors can vary very vastly between different cells, and we can use them as some kind of mathematical object that describes a cell. And suppose we can collect many, many different vectors from many different cells that we are interested in, and this is experimentally possible. This is a technique called single-cell RNA sequencing, and which I will not go into the technical details of, but eventually what we get is this very large matrix of gene expression for every cell that we measure in our culture. And we can ask many different questions, many interesting questions about this matrix, but one that would be particularly interesting in our context would be what is the level of coordination between pairs of genes in our genome? And why is this relevant? Because if you think a bit back about the illustration that I showed you before about this disrupted, or maybe our hypothesis of this disrupted state, then we can think of these cells as randomly expressing genes in a very uncoordinated way, and then we would expect to see a lower level of coordination once we measure this property of the matrix. And it turns out to be not very trivial to calculate this quantity. And to address this, we turn to a very well-known technique from data science and statistics called principal component analysis, or PCA. And I'm sure some of you in this room have heard about it before, but in any case, I'll give you an idea of what it is. So the idea of PCA is to look at our data, which in our case are just vectors of these cells, and to understand what would be the directions that span the data, okay? So in the case where the data is completely random and uncorrelated, what we get is that these arrows which represent the PCs, or principal components, are uniformly distributed, while in the other case where we have correlations and structure in the data, then they are distorted. Some of them become stretched while others are contracted. And then when we have our biological data, which contains thousands of dimensions and obviously cannot be plotted and visualized with our eyes, we can actually plot a histogram of these PCs, and where we have just the amount of arrows that correspond to a specific length. And this is what we typically get for our kind of data. And now what we can do is we can compare this to what we would get from a completely random matrix. And as you can see here, we get these bins of arrows that are longer than what we would expect from random, and also these bins that are shorter than what we would expect from random, which actually represent exactly this structure of stretching and contracting of the principal components. So this would be a way to identify these coordinations between genes. And also another interesting property is that what we see, which is maybe a bit counterintuitive, is that the bulk of these PCs, or the majority of them, actually fall within this regime of a random matrix. Yeah, so what I've done here is pretty controversial, right? I've compared biological data to random matrices, and but it turns out that this comparison teaches us a lot about the biological system, maybe much more than we would expect. For instance, we see that the data from the disrupted cells is much more comparable to these random matrices, meaning they exhibit more noise and less gene coordinations. And maybe even more interestingly, we understand that once we kind of peel off these layers of biological significance, what we get is this gene network that is kind of what defines these gene correlations that appears to be random. It's like peeling an onion and finding that the core is just a random matrix. And to kind of complete this picture, I just want to talk about the outer layers of this onion, which I stress are actually represent the biological pathways, okay, so what are these pathways? These are basically just these clusters of genes which are supposed to provide some specific biological function. And when we look at the cells that go after a regulated stress response, then we see that these pathways are activated very coherently in a very well-behaved manner. But on the other hand, when we look at cells that are disrupted, then we see that the activation of these pathways becomes less coherent, meaning not all of the genes that are in a specific pathway are expressed simultaneously. So this means that the pathways in the disrupted state are less coherent, are less relevant, and again, this is what reveals this inner layer of randomness. So to summarize, what I've shown you is that gene expression pathways and the analysis of pathways reveal these weaker coordinations in the disrupted state. And also, the underlying structure of gene expression can actually be represented by random matrices, even though in most biological cases, this is just screened by the more dominant biological pathways. And finally, we have learned that these disrupted cells, once we kind of remove the biological layers, reveal some very universal properties that can help us understand antibiotic resistance. So that is all. I would like to thank my lab members and collaborators, and of course, thank you all for listening.
תודה רבה, אלון. שאלות? Questions? Okay. Good.
Hi, thank you. Very interesting. My question is how you, from what you've showed us, how did you reach the conclusion that it's universal? That's my... and what exactly is universal? And,
yeah, okay, that's a bit of a philosophical question, but I think it's universal because this is something that is not a response to a very certain type of stress. It's something that we see in a very wide variation of antibiotics, of chemicals, of starvations, and it kind of shows us that the core of this bacteria in this case behaves like this. So I haven't proven it. It's not a rigorous
But
proof.
you did analyze. This really answers my question. So you analyzed a lot of stress ways to induce stress on these cells.
Yes.
Thank you. So maybe to continue Brock's question, at least in my field, when you talk about universality, you usually talk about some mathematical laws or I don't know, something that you can characterize in very different systems that emerge from different microscopic details but give the same mathematical law, for example. So is there something similar you can look for?
Well, I think it's analogous, but obviously you can't formulate exactly the same properties of universality in biology, but I think it's analysis just kind of because of the same reason, because the microscopic details here become irrelevant, because it doesn't matter if you've induced the stress by different manners and the final result is similar. But yeah, I don't think I can formulate a mathematical universality picture.
Thanks.
Thank you. Very interesting. So what was a bit counterintuitive for me is that when I think of stress and stress response. Stress response is many times very much regulated. Like if cells experience one type of stress, there are very specific gene programs that are turned on. So, what can you explain this discrepancy?
Exactly. I think that is exactly kind of the point. Like there is this regulated response that you are talking about, and but then there is also an option that the cell will not reach this regulated response. It will kind of be pushed away into a different regime, which is what we call the disrupted state. And I think sometimes it's kind of hard to say exactly when it will reach this state or this state, but usually it has to do with the how acute the stress is and how the dynamics of it are, like if it is very abrupt or if it is gradual, and this causes a different response of the cell. So this is a kind of a hypothesis, which is not proven, but what we believe is maybe that this is kind of a strategy in which the cell is exposed to something that it does not know how to deal with, and it kind of randomly tries to operate different mechanisms.
Maybe the other way around. I mean, not the type of stress that comes, but if a cell is at a certain state when the stress comes, it will behave in.
Yeah, that's also correct.
So you answered my question, but I'll ask a different one, maybe continuing with the previous answer. Do you think sometimes something that can appear random or very heterogeneous in a population is adaptive and regulated in the sense of maybe division of labor or bed hedging or some other adaptive value? Do you think this is the case here? Or is it
so that's a good question, and like you're saying, this could just be a case of heterogeneity when the population is being spread into these subpopulations. And this is not something we've seen, and so we believe this is not the case, at least so far.
Hi everyone, once again. So my title today is From Order to Disorder, and how transcription factors navigate complex genomes. I will tell you a bit about gene expression, about transcription factors, and in the end, if I have enough time, I'll share with you a small snippet of my recent work. So thanks to Alon, he gave a small introduction to what I'm going to talk to you about, but let's talk about the thing called gene expression and why do we need it, and what is it actually. So each cell in our body starts exactly the same, bearing the same genetic material. But somehow, in the end, it becomes a very specific cell, either a muscle cell or a nice cell, and it looks and functions differently. So this whole complex thing happens because of differential gene expression programs inside these cells. So what actually are genes? These are the small genetic materials stored inside our cells that affect not only a microscopic look of our cells, but also global features of our bodies, like the color of our hair or the shape of our fingers. But to talk about differential gene expression, I don't need these complex things like stem cell differentiation. What we can talk about is also about the small, simple organisms, just like yeast, and they need to somehow react to environmental cues and do it really fast. So I try to depict here a small baker's yeast, and in its proximity there is a heat source, and suddenly it needs to protect itself somehow. So what it does, it changes its expression program and produces proteins that will thicken its outer membrane, giving it an opportunity to survive the heat source. So the next question is, who are the key players in gene expression? First of all, this is the DNA, the genetic material stored inside our cells. The genes are kept inside it, and it's a linear stretch of letters, A, C, G, T, also called nucleotides, also called base pairs, and they are stored in chromosomes inside each live cell on planet Earth, each eukaryotic cell on planet Earth. The other key players are transcription factors, and I will focus mostly on them today. And these are proteins that know to navigate this DNA, find really small regions inside it. Then they can recruit their cofactors to mediate the expression of genes, either activate it or repress. But let's focus. There are also a very big field of nucleosome modification and epigenetics, but it is out of scope of my presentation today. So let's talk about transcription factors. Usually, not usually, but many proteins inside our bodies do actually have some specific shape, specific fold, what we call it. And if it disrupts, then the protein cannot exert its function. Transcription factors, on the other side, are not like this. So they do have a small folded part called DNA binding domain, denoted by the DBD here, the red blob. And this part is actually what finds the right regions on the DNA. The small stretches of 4 or 5 letters, 4 or 5 base pairs, like A, G, 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 G on it. But also, they have these huge non-DNA binding domain regions, which usually are intrinsically disordered. We call them IDR, intrinsically disordered regions. And they are like a tail. They don't have a fold. They just are there, either in front or flanking the DNA binding domain. And in it also reside activation domains or repression domains, which are responsible to recruit their cofactors. So in this linear view, the transcription factor here, denoted by these shapes, knows how to bind specific DNA sequence, like AGGGG here, and then using its activation domains, it can recruit coactivator, which will help to generate transcripts from this gene. And this, I think, exemplified, in my point of view, by the most classical example in yeast, by the GAL4 protein, which in regular conditions is repressed by GAL80, another protein that doesn't give the GAL4 an opportunity to work. And whenever a specific sugar called galactose enters the system, GAL80 can release the GAL4, and then the magic will happen. And the yeast will produce some proteins that will help to convert galactose into energy. So this linear model is okay, and it explains a lot of things, but transcription factors do something really serious, and they solve a really big problem in this process. So what the problem is? These small sites on the DNA, 4, 5, 6, 7 letters, happen in the genome 10,000 times, thousands of times. But transcription factors do bind only a small subset of them, 100, maybe 200. So there is a specificity problem. There are too many correct motifs in the genomes, but somehow transcription factors can and do find the right ones. And I try to depict it with this ball of string with the green motifs that are right and red ones, and somehow transcription factor has to detangle all of this and find the right ones. So transcription factors have different strategies to tackle this problem, but the members from our lab, from the lab of Naama Barkai and Weizmann, have showed us that actually the intrinsically disordered regions of the transcription factors, these tails, are those that grant specificity to transcription factors, which is really interesting. It was counterintuitive. And it's exemplified by this graph here. If you shorten the transcription factor gradually, more and more, it starts losing, the blue dots, gradually losing its specificity, meaning it still binds the correct motifs, but not at the right places. So what we conclude here is that IDRs, transcription factors, contain weak, globally spread specificity determinants. But, I remind you, these IDRs also contain activation domains inside them. So my question was, what if we fuse these small activation domains to the small DNA binding domains? What will happen in the genome? Will all locations that this minimal transcription factor binds be expressed or not? And we have explored this question in my recent publication from November in nucleic acid research. And if we focus on two groups of the DNA the transcription factor can bind, those that are shared between the full-length transcription factors and by the minimal DBDAD fusions, and those that are newly acquired, we can see on this graph that actually the regions that are induced, this green box on the right end, happen only when those two lines, the gray and the brown, collide together. And the gray line is the binding of the full-length transcription factors, while the brown line is of the DBDAD fusions. So somehow, only those areas that in native conditions, in regular conditions, are bound only by the full transcription factors, only they are induced by the minimal ones, leaving all those targets that the DBDAD bound not activate. We performed many more experiments, and in the end we found out that actually it's the coactivator who decides which genes will be activated or not. And this specific coactivator called MED15, this green blob here, knows how to find its right locations, although it doesn't have a DNA binding domain. So somehow it knows how to get the right places. I'd like to share with you more details in the questions if you like, and I would like to acknowledge my advisor, Professor Nama Barkai. Without her, I wouldn't be standing here today, and my colleagues who helped me to finalize the work, and of course Adam's Fellowship Program for an opportunity to stand and talk in front of you today. And I'd like to get some questions if there are.
Thank you, Vladimir. Questions?
Maybe I missed it, but what defines the correct binding sites in the genome? Because if they have the same code, then...
Yeah, so the motif essentially is the same for the correct, the small stretches on the DNA are the same for the correct sites and incorrect sites. But actually what we see is that nucleosomal architecture in the promoter is what decides if it's a right promoter or not. Somehow these IDRs, these floppy tails, know how to either recognize or interact with the right architecture, and then the magic happens. So we don't know yet. Yeah.
Very interesting, thank you. I know that the intrinsically disordered domain are considered to be responsible for formation of liquid-liquid phase separations, which are hypotheses to lead to gene expression. So could it be that this is actually what leads to the specificity? I mean, you need certain amount of occurrence of the DNA binding motifs to be in close proximity in the 3D organization matter, and only then the intrinsically disordered domain could lead to the formation of liquid-liquid phase separation, which will drive separation. So the question, to be more focused, is whether the definition of kind of the correct occurrence of a motif and an incorrect is the existence of many other motifs in its area that will then lead to liquid-liquid phase separation by the intrinsically disordered domain.
Okay, thank you for the question. So you're right in the sense that sometimes the correct promoters have a lot of occurrences of the right motifs, but sometimes it is not the case. And even one motif is enough for you to function. If you take, for example, the GAL4 transcription factor, the classical ones, so its core promoter doesn't have a lot of motifs inside it, and also its motif is very degenerate. It's like 3 nucleotides separated by 15 or 16 nucleotides and more 3 defined, so somehow...
But maybe in the 3D organization and not in the
yes.
promoter area.
So one could argue that in mammalian genomes it would be a really nice model because enhancers is a thing, but what I showed you is happening in yeast, where there are no known topologically associating domains to which you're referring. So it's a good hypothesis, and we'll have to explore it. but we still don't see a clear indication for this, because even without phase separation, somehow the expression does occurs.